So basically what you have here is, in your syllabus, is genetic engineering enzymes. You have genetic engineering enzymes. That is basically what you study here. And among the main genetic engineering enzymes are enzymes which are responsible for modification, cutting, pasting. So these are the enzymes which are responsible basically for modification, cutting and pasting. So as the definition of genetic engineering goes, you use certain cut paste techniques. Cut, paste, construction and destruction techniques and what do? You create a, rather than using recombinant DNA, you use genetically engineered product. Now this by genetically engineered product, you mean it could be recombinant DNA, it can be a recombinant protein. So basically, genetically engineered, or you can have a recombinant enhanced organism. These organisms, they may have a gain of function or they may have a loss of function. They can gain some function very well. Say for example, some proteins can express in much greater detail, or there may be a loss of function. Some proteins, when expressed too much, cause harmful effects, some cancerous drugs, so you can lose it. So, in the basic enzymes, what you mean is in terms of DNA. In terms of DNA, the basic enzymes are cut plus paste. So you have a particular circular DNA known as plasmids found in bacteria. Normally, they carry antibiotic resistance. And they are extra chromosomal DNA, they are circular in bacteria. Bacterial are prokaryotes, which means they don't have nucleus. They have a single nuclear DNA. If you have a look at a bacteria, you can have a nuclear region. Outside you can have the cell wall, and this is the nuclear DNA. This is sometimes this is called also as their chromosomal DNA. It is not in a two-sex chromosome like in eukaryotes. Now, apart from that, there are plasmids. Now you can isolate these plasmids outside. You can use special enzymes and you can cut them and linearize them. You can cut and linearize. And not only cut, you cut them at specific sites. And for example, you take another from another organism, you take another DNA. DNA from another organism. You take another DNA from another organism, and then what you can do with that is you paste them inside. And this process is known as ligation. Now, let us consider about what is this cutting and pasting enzymes, and you have some enzymes to modify. Just by cutting, you need to do some modifications. Just by cutting pasting is not enough. You need modifications which need to be performed. And what are these modifications? First is, let us talk about modifying enzymes. The first modified enzymes are nucleases. So nucleases, what do? They degrade nucleic acids. They degrade nucleic acids by breaking phosphodiester bonds. So nucleotide 1 is attached to nucleotide 2 by phosphodiester bonds. So they cut these bonds. They degrade nucleic acids by breaking the phosphodiester bonds, the phosphate bonds between nucleotides. Then, most important of them, which I talked about here, are the restriction endonucleases. Most important are the restriction endonucleases. They cut DNA at specific sequences. For example, one restriction endonuclease known as EcoR1, isolated first from E. coli, it cuts the sequence whenever there is a GAA-PTC and in the opposite direction, the reverse complementary. If this is 5' to 3', this is 5' to 3' in this direction. It always cuts in this. So always we will be having an overhang like this. And So it cuts. So whenever in a to put DNA, wherever you will find a GAA-AT site, EcoRN1 is going to digest it. Provided there is no modification like methylation or any other DNA modification done. If it's GAA-ATC, it's going to cut. Then the question comes that if EcoR1 digests all DNA at GAA-ATC, why does it not cut its own DNA? That also has got EcoR1 sites. There are also GAA-ATC sites. That is the question. Why does it not do? Because they have certain modifications. So there the EcoR1 sites are methylated. Sites are methylated. So it does not digest its own DNA. It does not digest its own DNA. And why should these enzymes exist in EcoR1? Basically, they are for countering viral attacks. So what will the virus do? They either pump in their DNA or RNA inside the host, and then they use the host machinery, transcribe, translate proteins, and they cause damage. They break, lyse open the proteins, and after they lyse open the proteins, the cell dies. Or they may keep on living like a parasite inside the particular cell, bacteria. Eventually, the health of the bacteria is not good. So they have got two phases, lysogenic phase where the virus keeps on living and living, or they have the lytic phase where when they have to propagate, they break open and new virus particles they are spread around. It's always happening correctly in the atmosphere outside us. Our body is fighting. There are several cells dying, several cells being born, several viruses being attacked. In human body, we have a mechanism known as interferons, which fight. That particular virus infected cell, it releases some chemicals known as interferons, which sort of warns the neighboring cells that there is a viral attack over here. It destroys itself, but the other cells, because of this interferon effect, they don't allow the virus to enter. Few of them die in the process. So essentially, these were to prevent the virus DNA. The moment the virus DNA comes inside, the EcoR1 is going to digest it. EcoR1, there are hundreds and thousands of restriction enzymes. Almost every sequence in this world, there is an enzyme for that. They are going to be released and they will digest that DNA. Or if it's an RNA, if the RNA comes inside, it's going to be converted into DNA. Then the moment it's converted into DNA, it is going to be destroyed totally, completely. No questions asked there. But human beings, they decided to use this particular property of cutting specific sequence to develop a process known as genetic engineering in order to make recombinant DNA. So you can have a human insulin gene taken from one of my cells. It's isolated. There are mechanisms known as polymerase chain reaction. You can amplify them, take them, and cut one of these plasmids with restriction enzymes, then use one of the enzymes known as ligase and ligate that foreign insulin inside bacteria. And then you can transform this particular plasmid with human insulin gene into a bacteria once more. Now the bacteria is starting to make human insulin. So all the insulin which is sold in the market is all not made from ox pancreas. During my grandfather's time, we used to use ox pancreas insulin. It's not used nowadays. It is only all genetically engineered. It's recombinant DNA insulin. Very easy to make. I mean, you can make it overnight. Tonight you inoculate bacteria. Next morning you get lots of tons of cells. They are grown. Lyse open the cells, pass them through a column. Immediately you have lots of insulin. You just simply package it. Of course, you have to do tests to understand what is the enzyme activity. I have done a lot of these. I have made a lot of insulin. I have made lots of other enzymes also. These are the parts of the modifying enzyme. The first one was the nucleus. The most important one was the restriction endonucleus. Okay. Endonucleus, the word, there are two types of words you have to use. Endonucleus and another is exonucleus. There are two types of nucleases. Endonucleus, if this is a double-stranded DNA, it cuts from the middle, cuts inside, cuts and digests inside. Inside DNA strands. And exonucleus, we have 5 prime, 3 prime. This is a double, it will cut from the exterior. It will keep on chewing the DNA. So you can divide them into two types. The 5 prime exonucleases, it cuts from the 5 prime side, and you have the 3 prime exonucleases. It cuts from the 3 prime side. So you can use them. So for a few names, it's good to understand few names. BAL31, it's a nucleus. You have exonuclease 3. Then you have deoxyribonuclease. Very famously used, it's known as DNA. Anywhere you want to remove DNA from any molecule, you use DNA, it's gone. Then you have S1 nuclease. This S1 nuclease is an endonuclease. Restriction endonuclease. In the mode of action, we are talking about nuclease BAL31. So it is a complex enzyme. It is a fast-acting 3' exonuclease. This is it, fast-acting. Very fast, it is true. And it is coupled with a slow acting restriction endonuclease. It's exactly the word restriction comes from, it restricts bacteria, the viral DNA which comes in the bacteria. So not only it is an restriction endonuclease, it's a very fast acting exonuclease, 3' exonuclease. Both 2 in 1 activity, but this is not very popularly used. So what it does, it cuts it's very fast, it chews away the DNA. You cannot use the BAL31 sequence to cut it. Then you have the exonuclease 3. Basically, I never preferred to use the BAL31 because I could not control the reaction very fast. I needed to shorten, say, if it's 100 base pair, I needed to shorten it 20 base pairs. I keep it for 30 seconds. It's standardized, but somehow this was very difficult to control. So I need something where I know after 2 minutes this much amount is going to be digested. I know something where it is very characterized, very difficult to do it. Former times they did it because it was isolated from bacillus strain. Achha, then exonuclease 3 is also a 3' exonuclease. So it will keep on chewing the 3' end. So if you have a double standard DNA strand, 5 prime, 3 prime, 5 prime, 3 prime, and you give them after some time, it's a 3 prime. It will act here, it will start to chew from here, it will start to chew from here. So gradually, you will have essentially a 5 prime overhang. So this region, this is the 5 prime overhang. This is also a 5 prime overhang. Same thing happens with exonuclease 3. You can use any of these enzymes. That's pretty well. Next important one is DNase 1. DNase 1 it cuts either single strand or double stranded DNA essentially at random. It cuts single or double stranded all these former enzymes are cutting single stranded 3 prime or 5 prime it was cutting it on that direction, but this is single or double stranded randomly যেখানে পাচ্ছে phosphodiester bond কেটে যায়, and this is the best enzyme to remove DNA traces. You are isolating RNA or protein, you need to remove DNA, RNA, so you put in DNA, that's it, gone. Then you have nuclear S1. What does nucleus S1 do? It subspecific for single-stranded RNA or DNA. It is specific for single-stranded RNA or DNA. So if you have single-stranded RNA, you can use S1 nucleus which could be, or single-stranded DNA which is present, like microRNAs which are present, it will be extended. Now we come to the next group of modifying enzymes. They are known as polymerases. We are now talking about polymerases. As the name says, it synthesizes. Now we are talking about destruction, or now we are talking about construction. So they synthesize copies of nucleic acid molecules. Simple definition: synthesize copies of nucleic acid molecules. And nucleic acids are either DNA or RNA. Then there may be two types. One is known as DNA dependent polymerase. The second is the RNA dependent polymerase. Second is the RNA dependent polymerase. So this is it depends on whether these polymerase they use one DNA strand as template, one DNA or one RNA as template. One single DNA or RNA strand is required as template. One single DNA or RNA strand is required as template. So on top of it, if it is DNA, then this enzyme, it sits and it synthesizes its reverse complementary sequence. The polymerase synthesizes its reverse complementary. For example, if you write 5 A, T, T, C, T, A, G, C, T, A, for example, the reverse complementary will be, this is the forward is 5 prime to 3 prime, the reverse is, this will be 3 prime to 5 prime, that is reverse, and complementary means opposite to C comes A, opposite to C comes A, G, A, T, C, G, A, T. So this is reverse complementary. This is what we mean by reverse complementary. So, a DNA dependent polymerase, one type of DNA dependent polymerase is DNA dependent DNA polymerase. We commonly call them as DNA polymerase, which we study in DNA replication. This is the DNA dependent DNA polymerase. So, as you know, it requires DNA as template. So, you require the and whatever you need NTPs, DNTPs, and other ions which you require. And you can also have an RNA dependent RNA polymerase. You have an RNA dependent RNA polymerase, and that requires RNA as a template, but it is RNA dependent DNA polymerase. I am crossing this out because for one reason. RNA dependent DNA polymerase. It will synthesize, this is an RNA, it will synthesize a reverse complementary DNA strand. DNA strand. Now, when you talk about RNA polymerase, it will synthesize reverse complementary RNA strand. They will synthesize a reverse complementary RNA strand. And now you can clearly say that this is synthesizing RNA. This reverse RNA dependent RNA polymerase this is sometimes it's a very recent finding. It is known as PARP. PARP. There was a paper named PARP ghost transcription. So these enzymes they synthesize nucleic acids by joining together nucleotides whose bases are complementary to the base template of the others. Like I said, they synthesize nucleotides. Nucleotides is a series of nucleic acids that we synthesize nucleic acids by using nucleotides. Adenine is the base. Adenine plus the sugar. Adenine, we say it's a base. Adenine plus sugar, it is known as adenosine. Adenosine plus sugar plus phosphate. Adenosine triphosphate. A nucleotide, nucleoside. This is a nucleotide. This is a side, nucleoside. So basically, you have the base attached. Base is attached to the sugar, and this is attached to phosphate, 3 phosphate molecules, and the phosphate goes like one more phosphate comes over here. O phosphate. Like this, you have another 3 phosphates, triphosphates. If we go for the sugar part. So they synthesize nucleic acids by joining together nucleotides. Nucleotide 1 plus nucleotide 2. So basically, I am talking this as N is a nucleobase. I am calling them N1, N2, N3, nucleotide, phosphate. You have a O oxygen molecule also. I am not drawing the oxygen molecule. So they synthesize phosphodiester bonds. Now any enzyme requires energy. Where does the energy come from? Now if you look at the structure of a phosphate, it's highly negative. It is attached to the sugar. these are high energy bonds like ATP. Sugar, and here is the base. If there is an adenosine, then it is adenosine triphosphate or ATP. If there is D-adenosine, it is D-adenosine triphosphate. In DNA you have D-adenosine and in RNA you have adenosine. So, this is high energy bonds. So, if you flip this part, a lot of energy is needed. And say this is the 5' part of the sugar, it will attack the 3' OH of another. Or another nucleic acid. And then this phosphodiester bond is formed. This is going to be removed. These two phosphates will be removed. PPI, pyrophosphate, this will be removed. And a lot of energy will be released. Will be released. A lot of energy will be released. So they synthesize nucleic acids. They join. They do a polymerase. Yeah. So the synthesis always occurs in the 5' to 3' direction. Five prime to 3' direction. So if this is the 5' side, this is the 3' side, 3' OH is over here. NTP comes, the 5' it attacks this part and gradually so this is N1PP. So it forms. Then again another 5' N2PP comes, attacks this. So the synthesis increases in the 5' to 3' direction. Synthesis starts to take place. This is how this progresses, this reaction progresses. And the actual synthesis is that the 3' end, 3' OH, in a sugar, for example, in glucose, if you take the structure of glucose, this is the first carbon, this is the second carbon, and this is the third carbon. In ribose sugar, this OH, this is the OH which is base target. In ribose, this is hexose 6. So in ribose, you will have here, say for example, you have the base attached. No, this is OH, H. Then you have the triphosphate going. This is the 5'. So this is the 3' OH. In DNA, it is H. In RNA, you have a OH present. This is how the ribose goes. So basically, the target, another 5' end is attacking this one, and the molecule starts to keep on increasing. So the requirement of the synthesis is you need to have a free OH. So already there must be a little bit of a primer. It just cannot synthesize on its own. You need a primer. Normally it's an RNA which provides that OH. Then DNA polymerase can keep on adding. It just cannot start adding from point 1. And this primer there is an enzyme known as RNA primase which fits it. This is also modifying enzyme. Okay. So DNA polymerase 1, it's a very হয়ে গেছে? So DNA polymerase 1, so the most famous polymerase is the DNA polymerase 1. Discovered by Conver. Discovered DNA polymerase 1. So it has got 3 types of function. Polymerase 1, 3 units are there in this. One is the 5' to 3' exonuclease. Suppose that the DNA is being replicated in a cell. Why do you? Because the cell is replicating, the DNA needs to be replicated. Suppose the cell is being replicated inside a cell. The DNA is being replicated. So one error can lead to a different protein. It can be lethal for the cell. So this has to be absolutely perfect. So there is a proofreading function. অ্যাডেনিনের উল্টো দিকে থাইমিনই আসতে হবে, মানে গুয়ানিন আসলে চলবে না। It can lead to an error. If that problem happens without proofreading, it's an error. So the 4 different types of ATPs are present. Say DATP, it can be DATP, DCTP, DPTP, DGTP. So there are 4 different mixes. So randomly, any one of them can come in that place. So the space is good enough for just a complementary one. এর উল্টো দিকে শুধু কি বসতে পারবে, অন্যটা ফিট করবে। বাই চান্স যদি ফিট করে যায়, বাই চান্স if it fits, then it is going to be replicated. The next cycle, this G will come instead of a T, and you can have a very lethal mutation. The protein may not be made. Dead. So it needs to be perfect, and this proofreading, when the moment that these exonucleases, they keep on scanning the DNA. The moment when there is a small problem, যে এটা ঠিক ঠিক যেরকম হওয়া সেরকম হচ্ছে না, কনফার্ম হাত বুলিয়ে ঠিক বুঝতে পারছি না, ঠিক লাগছে না, it is not plain, something is coming, there is a problem, remove it, কাট করে কেটে দেয়, কাট ওই কেটে কেটে ওইদিকে চলে যাবে। ধরো স্ট্যান্ডেতে এখানে একটা এরর এলো, এখান থেকে কিউ করে নিয়ে চলে যাবে, দিয়ে আবার সিনথেসাইজ করবে যতক্ষণ না ঠিকটা হবে। So that you have these exonuclear activities. So 1 in 100, 1 in 1 billion is the chance of an error in a perfectly proofread DNA polymerase. Although the tough polymerase, the PCR enzyme which we use at high temperature, it's got a very high PCR mutation rate, 1 in 10,000. That's bad. It has got a 3 prime too. Exonuclease plus polymerase, phosphodiesterase activity আছে। This has got also polymerase activity. So DNA polymerase 1, this is the famous. Many it is there are many techniques this is used for. One technique which is used is known as unit transcription, which someday I can explain to you, but not today. It's not in this part. So sometimes this becomes a big, very bulky enzyme with so many activities. You need to shorten it. So you need to have the polymerase and 5 prime, 5 prime, there is a 5 prime, it will also destroy it from both ends. So it's not needed. When you are using it for genetic manipulative purposes, not for natural causes. So you need to small it, shorten it. You can do it. We synthesize it as a, it's known as nanofragment. Normally, this is used for molecular biology studies, and this has got the this 5 prime 3 prime exonuclease activity is removed, so it retains the only the 3 prime to 5 prime exonuclease plus polymerase. দরকার নেই, এতগুলো দরকার নেই for proof reading. Just one is good enough. The 3 prime, we keep the 3 prime. 5 prime টাকে সেফ রাখো, 3 prime থেকে মারো। This nanofragment is needed where a single-stranded DNA molecule needs to be copied. তোমার এখানে ডাবল স্ট্যান্ডার্ড ডিএনএ দাও, এটাকে কপি করছো না, there you have a single stranded 5 prime 3 prime, and if both the ends of the 5 prime 3 prime এদিক থেকেও চুপ করে নেবে, এদিক থেকেও চুপ করবে, so basically it's going to disappear. So you need to remove the 5 prime side. Okay. DNA polymerase 1 তো করে দিলাম, প্লেনও হয়ে গেল। There is another important polymerase known as T4 DNA polymerase. So this is a template dependent DNA polymerase. T4 DNA polymerase is a template DNA template dependent DNA template template dependent DNA polymerase, and it catalyzes 5 prime catalyzes 5 prime to 3 prime synthesis from prime single stranded DNA. Prime মানে primer দেওয়া হচ্ছে, so right. This has 3' 5' exonuclease activities, but it does not have 5' 3' exonuclease activity. You don't need this 5' 3' nuclease activity. Or single stranded is more dangerous. Not so good. So this has 3' to 5' activity, no 5' to 3' exonuclease activity. It's like Leno fragment. You can use Leno or you can use T4 DNA polymerase. Because it's got a stronger 3' to 5' exonuclease activity of single stranded DNA. Stronger 3' to 5' exonuclease activities on single stranded DNA. And it has got weaker, it's weaker in double stranded DNA. Weaker in double stranded DNA. So what is the use? Use blunt ends. Blunting of ends, sorry. Blunting of ends, that is by filling the 3' or 5' overhead. ওভারহ্যাং वाला জিনিসটাকে তুমি ব্লান্ট ডিএনএ করতে ডিএনএটা এরকম আছে there is one overhang or one overhang you need to fill ends এটাকে পলিমারেজ দিয়ে লম্বা করে ব্লান্ট করে এন্ডস গুলোকে ব্লান্ট করে দেওয়া যায় এটা দরকার হয় অনেক সময় কাটিং টেস্টিং এতে and for example in such a DNA you can add radio-labeled nucleotides, so the ends of the DNA become labeled. So you can use this radio-label to probe something inside a cell. This part is radio-labeled, so you are probing this, putting it inside a cell where a complementary sequence will bind, and you can detect where the radioactivity is. It's needed for labeling to detect. ওকে আজকে এখানেই থাকুক হ্যাঁ অনেক আছে